Hi, my name is Monica Tjin, a research fellow working with Professor Carl Chikfason at Duke NUS Medical School, Singapore. Thank you for joining me in this webcast on our published work describing a method to culture human epidermal keratinocytes in a biologically relevant, chemically defined, and fully human system by using laminin for treatment of burns of skin injury. This study is a collaborative work between Duke NUS Medical School and Singapore General Hospital. I'd like to share a little bit about burns and burns treatment. Burns is a global public health issue causing 180,000 deaths annually. Burns typically occurs in low or middle income countries and mainly occurs in the African or Southeast Asia regions. In Singapore, being strategically located in the Southeast Asia regions, has one of the best burn centers that treats burns patients locally as well as from the neighboring countries within the region. And 10 to 15% of these patients suffer from major burns, and up to 30% of these patients requires donor skin grafting. So treatment for major burns can be done through split thickness skin autograft, where the healthy donor site is actually harvested and meshed to create a larger coverage before transplanting it back to the wound area. However, this method is not suitable for larger burns patients where there is a limited donor site availability. Cadaveric skin can also be used, but only as a temporary coverage, and it will get rejected after 2 to 3 weeks. However, the availability of these human donors for the allograft supply can also be the limitations. So due to all these problems, a cultured epithelial autograft emerged as the potential options due to its advantage in terms of minimum donor site mobility, minimum surgical trauma, and also it can be used for patients who has other medical issues such as diabetes, which might lead to non-healing donor sites. So cultured epithelial autograft, or CEA, is first developed by the late Howard Green 4 decades ago in 1975, where he discovered that human epidermal keratinocytes can be clonally cultured with the help of murine 3D fibroblasts as feeder layer. The particular mouse cell lines that work until now is the 3D3J2 lines, and it is still used globally for skin culture in the clinics. The culture process begins with preparing fresh mouse feeder cells, which is essential for growing skin cells, and this has to be irradiated to arrest their growth while maintaining their paracrine activity to stimulate the human keratinocytes growth. And this is followed by the keratinocytes culture process for about 3 weeks, and you can see here the colonies of skin cells supported by the adjacent feeder cells. So in Singapore itself, the CEA is currently an approved indication for patients with deep dermal or full thickness burns comprising a total body surface area of 30% and above, and also for critically ill patients with large wounds. However, even though it has been used globally for 40 years, this method is not without problems, especially for cell therapy where GMP standard on the cultured cells is expected. So the current CEA is categorized as an undefined system and classified as a xenograft or a high-risk product due to the presence of murine feeder cells in direct contact with the human cells, and also the presence of restricted biological agents such as chlorotoxin, as well as the chemically defined, undefined FBS, and therefore it increased the risk of the pathogens and immunogenic agents transmissions. On top of that, the manufacturing process is complicated as time coordination is required to prepare murine 3D fibroblasts in time before human skin cells is isolated. With that, there's a need to irradiate the feeder cells, and there are more characterizations and checkpoints for advantageous tests for animal viruses, etc. And also to ensure little trace of murine cell presence at the end of this process, which can only be done qualitatively. So what can we do to improve the system? So a few years back, Carl Chikfason, my boss, discovered that human recombinant laminin, particularly laminin 521, is able to replace mouse embryonic feeders for culturing embryonic stem cells. And ever since, it has been used for culturing both ES and also iPSC cells in xenogenic-free and chemically defined condition. So he has since founded BioLamina to produce these recombinant laminins. So we thought, why don't we apply the same concept for culturing keratinocytes? So before I go to that, I would like to explain to you what are laminins. So laminins are a family of extracellular matrix basal membrane proteins that appeared in multicellular organisms 500 million years ago. They are tissue-specific and has a specific function depending on their locations in the body. There are heterotrimeric proteins containing 3 chains, alpha, beta, and gamma, forming at least 16 identifiable isoforms, which are present in human body. For example, in laminin isoform 511, there is one alpha 5 chain, one beta 1 chain, and one gamma 1 chain. So currently, many of these isoforms are available in their recombinant format and are produced in-house in our labs as well as commercially available from BioLamina. So specifically in the skin, the keratinocytes are positioned on the basement membrane, separating the epidermal and the dermal layer. So within this basement membrane, laminin 3T2 and laminin 511 or 521 are the most highly expressed laminin isoforms. And the absence of laminin 3T2 on these junctions leads to skin blistering disease called epidermolysis bullosa. So we first identify and reconfirm again what are the laminin chains that are present in human skin basement membrane by using immunostaining as well as RT-PCR and RNA sequencing. So we further identified it if the murine 3G cells express any laminin proteins that might explain why they were able to robustly support keratinocytes growth. So we found that through the immunostainings as well as our RT-PCR and sequencing, the laminin 3T2, 511, and 521 are the main laminin isoforms present in on the skin basement membrane. And additionally, for the 3J2 fibroblasts, they actually express mainly laminin 411 and laminin 421. So, and then after we identified these laminin isoforms, we use these laminins to culture human primary keratinocytes. As shown here, without coating, the keratinocytes can't grow, and this is also the same for laminin 111. So surprisingly, they did not grow on the laminin 3G2, although this laminin is specific to skin basement membrane. And on the other hand, aligned with our hypothesis, the laminin 511 and 521, which are present on the skin basement membrane, were able to support keratinocytes adhesions and growth. And also surprisingly, the laminin 411, 421, which are the endothelial laminins, were able to support keratinocytes growth. So these two, laminin 411 and 421, are the laminins that are expressed by 3J2 fibroblasts. And we thought that is probably the reason why the 3G were able to support the keratinocytes growth. So among these 4 laminins, we found that the 2 laminins that are consistently robust are the laminin 421 and 511, whereas the other 2 laminins showed inconsistency in maintaining keratinocyte culture, such that on some patients, the cells are grown in these 2 laminins went to senescence in the earlier passage. So then next, we look at the growth curve comparing the 2 laminin system in comparison with the 3G method, and we also look at the expressions of the KC keratinocytes basal markers as well as differentiation markers on these cells across different passages by both qPCR as well as FACS, and found that their profiles are very, very similar to the 3G system. And also we look at the colony forming efficiency, and they are very similar even on the later passage. And we also looked at the karyotype on both early and late passage and found no chromosomal abnormalities or translocations. And next, we further looked at the RNA sequencing and found that the cells cultured on laminin system express KC markers level that are almost identical to the conventional method. And looking at the gene expressions across 25,000 genes, they are nearly identical. And finally, we tested our system on a flood model on a nude mice as our in vivo functional assay to see if the cells cultured on our system were able to fully engraft and stratify. So the analysis of the graft take by DH&E and immunofluorescence showed that our laminin cultured cells were fully engrafted and stratified, comparable to our control, with the KU80 in green marked the presence of the engrafted human cells. And a deeper analysis on keratinocytes basal and differentiation markers also showed similarities between laminin system and the 3G conventional system. And importantly, we noticed the presence of gamma-2 chain laminin present at the junction of human keratinocytes and the mouse connective tissue, indicating that these engrafted cells functionally express its laminin GD2, forming an epidermal-dermal junction. So in conclusion, we have successfully established a robust GMP-compatible keratinocytes culture platform that is chemically-defined and also fully human system. And as a safer alternative, this technology has less restrictions, and it can be very potential for other skin therapeutic applications, such as on the less severe burns as well as chronic wounds. And we are currently pursuing a pilot clinical trial to determine the safety and the efficacy of the new CEA in small burns. So thank you for listening. If you have any questions, you can find me on LinkedIn or you can email me at the link provided here. Thank you.